we discussed up to the taxonomy and cell biology. Now today we are going to discuss nucleus and nucleic acids. You all know nucleus is the dynamic center of the cell brain, which was discovered by Robert Brown, and nucleus shows outer nuclear membrane, double layered membrane with perinuclear or internuclear space in between, nucleoplasm, nucleolus and chromatin reticulum. Regarding those nucleus, chromosomes and other things, we discuss some bits. First is nucleoprotein nature is observed in chromatin, chromosome, nucleosome, plasmosome. Plasmosome means nucleolus. So nucleoprotein nature is observed in chromatin because it is present within the chromosome because it is containing chromatin other substances. Nucleosomes are basically made up of nucleoproteins which are discovered by Audet. And plasmosome is nothing but nucleolus discovered by Fontana, and plasmosome or nucleolus also shows nucleoproteinous nature. So answer is 1, 2, 3, 4. So nucleoprotein nature is observed in chromatin, chromosome, nucleosome and plasmosome, which is not true in the case of nucleus. All plants are uninucleate is wrong. Here 2 is given, but not that, 1 is correct. All plants are not uninucleate. There are some plants without nuclei. There are some plants without nuclei is there. All plants are not uninucleate. There are some plants without nuclei. There are some cells which are binucleate, and there are some cells which are multinucleate. So there are some cells without nuclei. Cells without nuclei is also correct because sieve tubes and sieve cells contain, initial stages they contain nuclei, but next stages they don't contain nuclei. So all plant cells are uninucleate will be the wrong option. Now, diploid chromosomal number of a plant A is equal to the haploid chromosomal number of a plant B. The triploid chromosomal number of a plant C is equal to the haploid chromosomal number of a plant B. Identify the plants ABC. For this you have to know this tabular form. Haplopappus, the chromosomal number is 4. Pisum sativum, the chromosomal number is 14. This is diploid number. Allium, the chromosomal number is 16. Zeames, the chromosomal number is 20. Oryza, the chromosomal number is 24. Nicotiana, the chromosomal number is 48. Gossypium, the chromosomal number is 52. And Saccharum, sugarcane, the chromosomal number is 80. This tabular form is very important to do these type of problems. We have to write haploid is 2, triploid is 6. Like this for all the things, if you write this, the answer will be Oryza, Nicotiana, Allium. Diploid chromosomal number in plant A. Plant A, here Oryza. Oryza is 24. Diploid number of chromosome of a plant A is equal to the haploid number of chromosome of plant B. 24 half is 12. So haploid number of chromosomes in Nicotiana. Nicotiana is 48, half will be 24. Then the triploid number of chromosomal number on the plant C. Plant C here is Allium. Triploid number of chromosome Allium is 24 is equal to the chromosomal number of the plant B. B is just now we told Oryza. So for this the answer is Oryza, Nicotiana, Allium. So what is the question? Question is diploid chromosomal number of a plant A is equal to the haploid chromosomal number of plant B. The triploid chromosomal number of a plant C is equal to the haploid chromosomal number of plant B. Identify the plants A, B, C respectively is Oryza 24, Nicotiana 48, Allium is 16, but when it comes to triploid, 16 plus 8, 24. So answer is Oryza, Nicotiana, Allium. Which form of chromatin is genetically more active? Which form of chromatin is genetically more active? Chromatin is highly coiled thread-like structure. At some portions it is more coiled, at some portions it is less coiled. The more coiled portion takes more stain and appear dark. This portion is called heterochromatin. Heterochromatin is also called false nucleolus. The other name of heterochromatin is false nucleolus. The light stained portion which takes less stain, it appears light in stain. This portion is called euchromatin. Normally, active genes are present in euchromatin. Inactive genes are found in heterochromatin. So, according to this question, which form of chromatin is genetically active means lightly stained portion euchromatin has many active genes. Dark stained heterochromatin do not have genetically active gene. Euchromatin and heterochromatin is wrong. Neither euchromatin nor heterochromatin is also wrong. So, which form of chromatin is genetically more active means lightly stained euchromatin where the genetically active genes are present, and this is the region where the formation of messenger RNA occurs. That means transcription occurs, messenger RNA is formed, proteins are produced with the help of messenger RNA, everything occurs in the light stained portion that is called euchromatin. Select the incorrect statement regarding the structure of chromosome. Select the incorrect statement regarding the structure of chromosome. Prokaryotic chromosomes do not possess histones is correct. Prokaryotic chromosomes do not possess histone proteins is correct. A metaphase chromosome has two sister chromatids, correct, because every metaphase chromosome has two chromatids and 4 arms. So, a metaphase chromosome has two sister chromatids, correct. A nucleolar organizer is a secondary constriction. Any secondary constriction in a chromosome, not all the secondary constrictions, some secondary constrictions in a chromosome are regarded as nucleolar organizer. A telocentric chromosome has one chromatid with 2 arms. Telocentric means this one. Telocentric means an eye-shaped chromosome. It shows centromere towards the proximal end and it shows only one chromatid arm or only one chromatid and arm. So, telocentric chromosome is an eye-shaped chromosome which has one chromatid and 2 arms is wrong. It has only one chromatid and one arm. One chromatid and one arm. Kinetochores do not associate with secondary constrictions. In any chromosome, primary constriction will be at the center, and this primary constriction contains special protein discs called kinetochores. The same secondary constriction will not have kinetochores. So, kinetochores are absent. Because of the absence of kinetochores, the secondary constriction or at the point of secondary constriction, the chromosomes cannot bend. The spindle fibers also cannot attach at the point of secondary constriction because this portion do not possess protein discs called kinetochores. So, definitely in these 5 options, the incorrect statement is a telocentric chromosome has one chromatid and 2 arms. Telocentric chromosome, the centromere is at the proximal end. Chromosome shows only one prominent arm, one chromatid and one arm. It is an eye-shaped chromosome. So, answer is 4 alone. The length of DNA is 272 angstroms. If it consists of 20% of cytosine, how many hydrogen bonds are present? Here you have to do it 272 by 34. Why? Because 34 angstroms is one spiral. If you want to know how much 27 angstrom spiral means, 8 spirals. One spiral contains 10 pairs. 8 spirals contain 80 pairs. 80 pairs means 160. 20% of 160 is 20% of 160 is cytosine. That means in this 32 are cytosines. If 32 are cytosines, 32 are guanines. 32, 32, 64. Remove 64 from 160. 6 and 10 log 6, 9, 96. 96, 48 are adenines and 48 are thymines. So, 32 into 3, 48 into 3, 48 into 2. By this you will get the hydrogen bonds, and the hydrogen bonds, the question is the length of DNA is 272 angstroms. If it consists of 20% cytosine, how many hydrogen bonds? Answer is 192. How it will be done? 34 angstroms is one spiral. 272 angstroms means 8 spirals. For one spirals, 10 pairs are present. For 8 spirals, 80 pairs are present. 80 pairs means 160. 80 pairs means 160. 160 contains 20% of cytosine. That means 32 cytosines, 32 guanines. The remaining are adenines and thymines. The answer will be 192 hydrogen bonds. Which is not true regarding nucleolus? So, here there are statements given which is not true regarding nucleolus. First is nucleolus is covered by a membrane. Second is it has abundant RNA and traces of DNA. Third is its central part is fibrillar in nature and peripheral part is granular in nature. Fourth is it synthesizes ribosome. It has abundant RNA and traces of DNA is correct. Abundant RNA and traces of DNA is correct. Its central part is fibrillar in nature and peripheral part is granular in nature is correct. It is the place where ribosomes are synthesized. Nucleolus or plasmosomes are popularly called as ribosomal factories of the cells. That means ribosomes are synthesized in this. Now, regarding nucleolus, wrong statement is nucleolus is covered by a membrane. There are two cell organelles in the cells which are not surrounded by any outer membrane. They are nucleolus and ribosomes. These do not possess any outer limiting membrane. Only two cell organelle, one in nucleus, one in cytoplasm without any outer membranes. Nucleolus is the structure which produces ribosomes. Nucleolus itself is not having any outer membrane. So, the ribosomes that have come from nucleolus do not possess any outer membrane. So, the answer for this is nucleolus is covered by a membrane is wrong. Certain secondary constructions of chromosomes in the nucleus have the genes of formation of nucleoli. Certain secondary constructions of chromosomes in the nucleus have the genes for the formation of nucleoli. This is the assertion. The secondary construction of all chromosomes in the nucleus are called nucleolar organizers. First one is correct. Certain secondary constructions of chromosomes in the nucleus have the genes of the formation of nucleoli is correct. But the secondary constructions of all chromosomes in the nucleolus called nuclear organisms is wrong, because all secondary constructions cannot be called as nucleolar organizers. Only few are called as nucleolar organizers. So, answer for this is A is true but R is false, because certain secondary constructions, certain is used here. Secondary constructions of chromosomes in the nucleus have the genes of formation of nucleoli is absolutely correct. But the secondary constructions of all chromosomes are capable or called as nucleolar organizers is wrong. So, A is true, R is false. RNA does not differ from DNA in. RNA does not differ from DNA in. In DNA and RNA, you will find pyrimidines and purines. In both, pyrimidines and purines. Pyrimidines are cytosine, thymine, purines are adenine, guanine. Here also, in DNA, pyrimidines are cytosine, thymine, purines are adenine and guanine. But in RNA, pyrimidines are cytosine and uracil. That means the thymine in DNA is replaced by uracil in RNA. So, purines are same, adenine and guanine are same in DNA and RNA. When it comes to pyrimidines, there is a little bit different, that is, in DNA thymine is present, in RNA uracil is present. So, RNA does not differ from DNA in type of sugar, definitely differ, because RNA has ribose sugar with C5H10O5, DNA has deoxyribose sugar with C5H10O4. Pyrimidines, RNA has cytosine and uracil, DNA has cytosine and thymine. Number of strands, some DNA has double stranded, some DNA has single stranded, some RNA has something like that. So, the difference lies in purines, does not lies in purines, because DNA is double strand, RNA is single strand. Question is, RNA does not differ from DNA in purines, because in both DNA and RNA the purines are adenine and guanine. The another name of nucleolus is plasmosome. Nucleolus was discovered by Fontana. The other name of nucleolus is plasmosome. Ergosome is the other name of ribosome. 1 and 3 are same. So ergosome is the other name of ribosome. Karyosome is the special structures present in nucleolus. So another name of nucleolus is plasmosome. Now, correct the order of arrangement of different parts of metacentric chromosome from primary constriction towards the polar ends. First is kinetochore. Next is, for this the answer is 1. First is kinetochore. Next is nucleolar organizer. Kinetochore is the protein disc present in centromere. Next is nucleolar organizer. Then is satellite. Then telomere. So from centromere, see, this is centromere, this is chromosome, this is secondary constriction, and this is telomere. This is centromere with kinetochore. This is secondary constriction. This is satellite, and this is telomere. So terminal end of chromosome with the polarity is called telomere. So order is kinetochore, secondary constriction, satellite, and telomere. Answer is 1. 1, 3, 2, 4. 1 is kinetochore. 3 is nucleolar organizer. Nucleolar organizer means secondary constriction. Then 2 is satellite, and last is telomere. What is telomere? The terminal end of a chromosome with polarity. Because of this polarity, chromosomal ends cannot fuse with each other. Polarity is clearly found in eukaryotic chromosomes. That's why eukaryotic chromosomes are rod-shaped in nature. The polarity is not found in prokaryotic chromosomes. That's why prokaryotic chromosomes are circular in nature. Metacentric chromosome from primary constriction toward the same question. 1, 3, 2, 4. Kinetochore, nucleolar organizer, satellite, and telomere. Nucleus is totally absent in these cells in the entire plant kingdom. In the entire plant kingdom, nucleus is totally absent. Xylem parenchyma is a dead shell. We cannot regard this here because here nucleus is absent. Then, since xylem parenchyma is dead, but it is the only living component in xylem which has parenchyma cells. So, xylem parenchyma out of xylem tracheids, xylem trachea or xylem vessels, xylem fibers, xylem parenchyma. The only living tissue is xylem parenchyma which contains nucleus. Then companion cells. Companion cells are the special parenchyma cells which are very closely associated with sieve tubes. Companion cells and sieve tubes both originate from the same parental cell. But the difference is sieve tube loses the nucleus at maturity, companion cell regain the nucleus at maturity. So, companion cell regain the nucleus means it definitely has nucleus. Mature sieve tubes of phloem, this is the correct answer. Mature sieve tubes of phloem are always enucleate. Enucleate means they do not possess any nucleus. Then young sieve tubes of phloem, definitely contain nucleus. So, xylem parenchyma contains nucleus, companion cells contain nucleus, young sieve tubes of phloem contain nucleus. The only component without nucleus is mature sieve tubes of phloem. So, old sieve tubes or developed sieve tubes or mature sieve tubes of phloem are enucleate or without nucleus. Answer is 3. Find the odd one with respect to the chromosomal number. Root tip cell of oryza. Just now I told about the tabular form. Root tip cell of oryza will be 24. Endosperm cell of onion. Endosperm cell of onion means that is allium. Allium is 16, 24. Pollen grain of tobacco. Tobacco is 48. So, half 24. So, pollen grain of tobacco is 24. So, root tip cell of oryza is 24 chromosomes. Endosperm cell of onion or allium is 24 chromosomes. Pollen grain of tobacco is 24 chromosomes. Egg cell of gossypium is 26 chromosomes. So, find the odd one in the respect to the chromosomal number is root tip cell is 24, endosperm cell of onion is 24, pollen grain of tobacco is 24. So, the egg cell of gossypium is 26. So, here the answer is 4, egg cell of gossypium. This is a matching tabular form. Karyotype, kinetochore, heterochromatin, ideogram. Answer is 3 because karyotype A is 3. Characteristic diploid chromosome complement of a species which gives shape, size, number and many other aspects is called karyotype. So, karyotype is 3A. Characteristic diploid chromosome complement of a species. Then B2, kinetochore, darkly stained regions. B2, darkly stained regions with inactive genetic materials. Heterochromatin, 1. Kinetochores are site of implantations of spindle fibers in chromosome. 2. B is 2, site of implantations of spindle fibers in chromosomes. Why? Because kinetochores contain implantations of microtubules. Whenever microtubules are present, spindle fibers are formed from microtubules. So, they directly go and attaches to primary constriction because of the presence of kinetochores. Heterochromatin, darkly stained region with inactive genetic material. Lightly stained region with active genetic material is euchromatin. Darkly stained region with inactive genetic material is heterochromatin. Ideogram. Ideogram is 4. Diagrammatic representation of diploid chromosome complement. That means diagrammatic representation of karyotype. So, a single set of chromosome here is called genome. On list 1, genome is not given. So, a single set of chromosome is called genome. So, karyotype is characteristic diploid chromosome complement of a species. Kinetochore is site of implantations of spindle fibers in chromosome. Heterochromatin is darkly stained region with inactive genetic material. Ideogram is diagrammatic representation of diploid chromosome complement. Answer is A3, B2, C1, D4. This is that matching. A DNA molecule with 408 angstroms. A DNA molecule with 408 angstroms length contain 40 adenines. Find out the total number of pyrimids. Just now I told 408 angstroms means 408 by 34. 408 by 34. 408 by 34 will be 12 spirals, 120. So, 40 adenines, the answer will be 120. Answer will be 408 angstroms means 408 by 34. 408 by 34 is 12 spirals. 12 spirals means 120 pairs. 120 pairs means 240. In this 240, 40 are adenines. 40 are adenines means the remaining 80 will be guanine. So, in total it is 120. Nucleus and chromatin are discovered respectively by, respectively means first who discovered nucleus, then who discovered chromatin. Robert Brown and Hofmeister, Robert Brown and Fontana, Robert Hooke and Hofmeister, Hofmeister and Fontana. Robert Brown discovered nucleus, Hofmeister discovered chromatin. That is the answer. Robert Brown discovered nucleus, Fontana discovered nucleolus or plasmosomes. Robert Hooke discovered cell and cell wall. Hofmeister discovered chromatin. Hofmeister discovered chromatin, Fontana discovered nucleolus. So, the nucleus and chromatin are discovered respectively by Robert Brown and Hofmeister. Identify the correct descending order of the RNA based on their molecular weights. Correct descending order means based on their molecular weight. Descending order means from higher to lower. The molecular weight of messenger RNA is 10 lakhs. The molecular weight of ribosomal RNA is 100 into 10 power 4 lakhs. And tRNA is 2.5 into 10 power 4. That means 2500 Dalton. So, 10 lakhs is this one and this 5 lakhs. 5 lakhs is mRNA, 10 lakhs is rRNA and tRNA is 2500. So, answer is 1. First ribosomal RNA, then messenger RNA, then tRNA. So, molecular weight of ribosomal RNA is 10 lakhs. Molecular weight of messenger RNA is 5 lakhs. Molecular weight of transfer RNA is 2500 Dalton. So, answer is ribosomal RNA, mRNA and tRNA. Answer is 1, 1, 3, 2. Find out the incorrect statement in the following. Incorrect statement. Complementary base pairing is present in adapter RNA. Adapter RNA means Adapter RNA means tRNA. Ribosomal RNA is synthesized in plasmosome. It is also correct. Complementary base pairs are present in adapter RNA is correct, because in tRNA only unpaired base pairs are CCA. All the other base pairs are connected by hydrogen bonds. Base pairs are absent in highly unstable RNA. Highly unstable RNA means messenger RNA. Here the base pairing is completely absent. Messenger RNA is interpreter of genetic code is wrong. Messenger RNA is an interpreter of protein synthesis and amino acids and other things. Okay. So messenger RNA is interpreter of genetic code is wrong. Complementary base pair is present in adapter RNA or transfer RNA is correct. Ribosomal RNA is synthesized in plasmosomes or nucleolus is correct. Base pairing is absent in highly unstable RNA. Highly unstable RNA is messenger RNA, because its lifespan is 2 minutes in prokaryotic cells, around 4 hours in eukaryotic cells. Backbone of DNA. Backbone of DNA. Any DNA has 3 parts: nitrogen base, sugar and phosphates. Nitrogen base are pyrimidines, purines. Pyrimidines are cytosine, thymine. Purines are adenine, guanine. Sugar is deoxyribose sugar with C5H10O4. Phosphates are attached to sugar. Phosphoric acid is present as phosphates and attached to sugar. Normally, sugars and phosphates in DNA are regarded as backbones of DNA. Sugars and phosphates and DNA are regarded as backbones of DNA, and these sugars and phosphates, the backbones of DNA, are connected by phosphodiester bonds. The nitrogen base of one strand, is attached to the nitrogen base of other strand by hydrogen bonds. The sugar and nitrogen bases are attached by glucosidic bonds, and sugar and phosphate, which forms the backbones, are attached by phosphodiester bonds. So the answer for this is 4. Arrange the following nucleic acids with respect to their gradual increase in their amount in a cell. Maximum amount is present in DNA. Gradual increase. So minimum is, answer is 3, 4, 3, 2, 1. So increase of their amount, gradual increase. Less is mRNA, 5 to 10%. Next is tRNA, 10 to 15%. Next is ribosomal RNA, around 80%. DNA maximum. So arrange the following nucleic acids with respect to their gradual increase in the amount in their cell. So answer is 4, 3, 2, 1. Messenger RNA is just 5 to 10%. Transfer RNA is 10 to 15%. Ribosomal RNA is 75 to 80%. DNA maximum. So mRNA, tRNA, rRNA, DNA. Answer is 3 for this. Arrange the following organisms in ascending order based on their sequence in chromosomal number. Nicotiana is 48. Man is 46. 23 pairs. Oryza is 24. Haplopappus is 4. So answer is ascending sequence, less to more. First Haplopappus 4, then Oryza 24, then man 46, then Nicotiana 48. So 4, 3, 2, 1. 3 is the answer. So Haplopappus 4 chromosomes, Oryza 24 chromosomes, man 23 pairs and 46 chromosomes, Nicotiana 48 chromosomes. Answer is 4, 3, 2, 1. Now this is a 3-column tabular form. Type of nucleic acid, nitrogen base present, function. rRNA. Type of nitrogen base absent, rRNA. In rRNA, guanine is present. It maintains ribosomal structure, wrong. mRNA, thymine brings protein synthesis information to ribosome. tRNA, cytosine brings amino acid to the ribosomal surface, correct. DNA, uracil. So here the 2 incorrect answers are 2 and 4, not correct. It is incorrect. Combinations is 2 and 4. So third option. hydrolytic enzymes are present in. Hydrolytic enzymes means the enzymes which are very active in the presence of water. They are present in lysosomes and nucleoplasm. Nucleoplasm is the site with proteolytic enzymes, hydrolytic enzymes, ribonucleoproteins and many components, and lysosomes definitely possess hydrolytic and phospholytic enzymes. So, lysosomes and nucleoplasm. Peroxisomes contain peroxidase and catalase. Lysosomes and peroxisome is wrong. Glyoxysome is wrong. Glyoxysome contains the enzyme which helps in glyoxylate cycle, that is conversion of fats into carbohydrates. So, answer is 2. Nucleolus develops from the following part of chromosome. Just now we explained. Nucleolus does not come from primary constriction. It does not come from telomere, polarized ends of chromosome with polarity. It does not come from centromere, because centromere and primary constriction are same. So, nucleolus comes from some of the secondary constrictions, because all secondary constrictions cannot be considered as nucleolar organizers. Only some secondary constrictions are considered as nucleolar organizers, which gives rise to nucleolus. So, answer for this, nucleolus develops from the following part of chromosome means secondary constriction. Answer is 2. Now, 2 correct combination: chromosome type, number of arms in anaphase, shape of chromosome in anaphase. Metaphasic to L-shaped chromosome, submetacentric to V-shaped chromosome, acrocentric to J-shaped chromosome, telocentric 1 I-shaped chromosome. Answer is 3 and 1, because metacentric is not L-shaped, it is V-shaped. Submetacentric is not V-shaped, it is L-shaped. So, the shape itself is wrong. Metacentric is V-shaped, submetacentric is L-shaped, but number of arms in anaphase 2 is correct. Acrocentric 2, one is a very small arm, the other is an exceptionally large arm. J-shaped is correct. Telocentric is 1, I-shaped is correct. So, the correct combination of answer in this 26th question: chromosome type, number of arms in anaphase and shape of the chromosome in anaphase is always 3 and 4. So, answer for this is third option. Now, this is a assertion and reasoning question. Plasmosome is present in dynamic state or dynamic center of the cell. Dynamic center of the cell is nucleolus. Dynamic center of the cell is nucleolus. Plasmosome is present in dynamic center of the cell is correct. Plasmosome is nucleolus. It is present in the dynamic center of the cell, that is nucleus. Ribosomes are present in nucleolus. Definitely nucleolus contain ribosomes. Why? Because nucleolus is the place where ribosomes are synthesized. So, plasmosomes are present in dynamic center of the cell is correct. Ribosomes are present in nucleolus is correct, but because ribosomes are present in the nucleolus, plasmosome is not present in the nucleus. So, A and R are correct. R is not the correct explanation of A. A and R are correct. R is not the correct explanation of A is the option for this. Another assertion reasoning. An acrocentric chromosome contains or attains L shape in anaphase. Acrocentric chromosome means it appears like this. Acrocentric chromosome means centromere present at the proximal end, one chromatid is present where one is a small arm, the other is an exceptionally large arm. So, it is called J-shaped chromosome, not L-shaped chromosome. So, an acrocentric chromosome attains L shape in anaphase is wrong. It cannot attain L shape, it attains J shape. It has a centromere just below the telomere. So, here it is telomere. Because the arm is very small, centromere is definitely present just close to telomere. So, assertion is wrong, reason is correct. What is assertion? An acrocentric chromosome attains L shape in anaphase is wrong. An acrocentric chromosome attains J shape in chromosome is correct. It has centromere just below the telomere is correct. So, A is false, R is true. Fourth option is for this. Another assertion reason. Purines and pyrimidines exist in 1:1 ratio in DNA. Purines and pyrimidines exist in 1:1 ratio in DNA is correct, because chemical analysis by chemical analysis on DNA by Chargaff. A famous scientist by name Chargaff conducted chemical analysis on DNA. So according to Chargaff chemical analysis of DNA, DNA contain pyrimidines and purines in 1:1 ratio. It is correct. The total number of adenine nitrogen bases is equivalent to thymine nitrogen bases in the same strand is wrong. The total number of adenine nitrogen bases is equivalent to thymine nitrogen bases in complementary strand is correct, not in the same strand. So purines and pyrimidines exist in 1:1 ratio in DNA is correct. The total number of adenine nitrogen bases is equivalent to the thymine nitrogen bases in the same strand is wrong. The total number of adenine nitrogen bases is equal to the total number of thymine nitrogen bases in the complementary strand. If it is given, it is correct. So here A is correct, R is false. Another assertion reasoning. Hydrogen bond formation between nitrogen bases is not seen in mRNA. Hydrogen bond formation in between nitrogen bases in mRNA is not seen in mRNA. Why? Because mRNA is single stranded and a long component without any foldings. Nowhere in mRNA you will find foldings. Whenever foldings are not seen, no question of formation of hydrogen bonds. No question of formation of hydrogen bond. It is very popularly called a long linear strand of RNA, which does not show any overlappings. Any overlappings or foldings. So that is correct. The lifespan of prokaryotic mRNA is 2 minutes. It is also correct. But because of the lifespan of prokaryotic mRNA 2 minutes, the hydrogen bond formation between nitrogen bases is not seen in mRNA is wrong. So both A and R are correct. R is not the correct explanation of A. So answer is 2. Fibrillar region and granular regions are seen in. Nucleus does not show fibrillar region and granular region. Cytoplasm does not show fibrillar region and granular region. Plasma membrane does not show fibrillar region and granular region. Nucleolus or plasmosome show outer fibrillar region and inner granular region. So fibrillar and granular regions are seen in nucleolus. Second option. Lightly strained regions of chromosomes are heterochromatin or euchromatin with active genes. Third option, euchromatin with active genes. Now, the total number of turns that the DNA makes around a core of nucleosome. Every core of nucleosome means H2A, H2B, H3, H4. This is the core of nucleosome. These appear in pairs, so it is an octamer. It is circled by DNA in 2 spirals. So an octamer of 8 histone proteins plus 2 supercoils of DNA. 2 supercoils of DNA becomes nucleosome. So according to this question, around the central core of H2A, H2B, H3, H4 in pairs, that means 8 histone proteins. The total number of turns that DNA makes around the central core of nucleosome is 2. 2 turns. First option. The part of DNA present in between 2 adjacent nucleosomes. Just now I told 1 nucleosome, 2 coilings of DNA. Next nucleosome, 2 coilings of DNA. The DNA that runs from one nucleosome to other nucleosome is called linker DNA or spacer DNA or internucleosomal DNA. So the part of DNA present in between 2 adjacent nucleosomes is called spacer DNA, it is called linker DNA, it is called internucleosomal DNA. So answer for this is all of the above. All of the above will be the answer for this. One of the following histone proteins is not an integral part of core of nucleosome. Just now I told nucleosome is a combination of pair of H2A, H2B, H3, H4. It shows supercoiling of DNA 2 spirals. and it will enter another central core with H2A, H2B, H3, H4. This is called internucleosomal DNA or spacer DNA. Wherever DNA is entering and coming out, a special protein is present on the outside which is called H1 protein. So the histone protein, which is not forming the central core of nucleosome, which is not seen in the central core of nucleosome, or one of the following histone proteins is not an integral part of core of nucleosome, is H1. So H1 protein is not seen in the integral part because H3, H4, H2A, H2B are found in integral part of nucleosome. Genetic nature of DNA was confirmed by, after discovering DNA, nucleic acids by Hershey and what you called Joseph Frederick Messer, Hershey and Chase conducted special transduction experiments. Hershey and Chase conducted special transduction. What is transduction? Transfer of genetic material of one bacterium into another bacterium with the help of a bacteriophage or virus. Transfer of genetic material from one bacterium to other bacterium with the help of a bacteriophage is called transduction. So transduction experiments are conducted by Hershey and Chase, and they proved that DNA is the genetic material. So Messer transduction experiment is wrong. Chargaff chemical analysis proved that DNA contains pyrimidines and purines and 1:1 ratio. That's why it is definitely doubles or two coils or what you call double helix. RNA does not possess 1:1 ratio of pyrimidines and purines, so it cannot form double helix. Franklin X-ray diffraction studies only helped to construct the DNA or propose a double helical DNA by Watson and Crick. So here the answer is Hershey and Chase, who conducted transduction experiments. A nucleoside has a nucleoside. There is a difference between nucleotide and nucleoside. Nucleotide means nitrogen base, sugar and phosphate. If phosphate is not there, only nitrogen base and sugar are there, that is called nucleoside. So nucleoside is nitrogen base and deoxyribose sugar without phosphate. So deoxyribose sugar, phosphate is wrong. Pyrimidine, purine wrong. Nitrogen base and phosphate is wrong. Nitrogen base and deoxyribose sugar without phosphate radical is called as a nucleoside. Answer is 2. Phosphodiester bonds are formed between, previously we discussed, sugar and phosphate forms the backbones of DNA. The backbones of DNA are connected by phosphodiester bonds, not phosphate and adenine, not deoxyribose and adenine, not adenine and guanine. In between adenine and guanine, hydrogen bonds are present. So answer is phosphate and deoxyribose. Deoxyribose sugar and phosphate always show phosphodiester bonds. The unpaired nucleotides present in the tail of transfer RNA. Every transfer RNA shows a clover leaf model like this. A clover leaf model proposed by Holle. It has one loop with anticodons. It has another loop which recognizes ribosome. It has another loop which recognizes ribosome nuclei or enzyme that activates amino acid. It has an accessory loop, and on one side tail always ends with CCA nitrogen bases. This is the place where amino acids are attached. So the unpaired nitrogen bases in transfer RNA are always CCA. Third option, the unpaired nucleotides present in the tail of transfer RNA are CCA. Pseudohelices are formed by, DNA forms perfect helices. Messenger RNA is single stranded, cannot form helices. Single stranded DNA absolutely cannot form helices, but ribosomal RNA forms pseudohelices. So pseudohelices are clearly formed by ribosomal RNA. So these are some of the questions in nucleic acids and chromosomes. Nucleus, nucleic acids and chromosomes. Now we discuss cell division.